In biology, the term nutritionally non-essential does not mean optional. It creates this totally false impression that you can live without them. The reality is the total opposite.
Go on.
These amino acids are so biologically critical, so fundamental to your minute-by-minute survival, that evolution refused to outsource them.
It refused to outsource them. What do you mean?
I mean that nature looked at these molecules and said, I cannot risk this organism failing to find this in a berry bush or a piece of meat. If we run out of this, we die. Instantly. So the body kept the blueprints. We maintained the factory floor to manufacture them in-house 24/7.
That completely flips the script. So non-essential actually means homemade.
Homemade is the perfect word. And the essential amino acids, the ones we see on supplement bottles and obsess over in diet blogs, those are actually the ones evolution just sort of shrugged at.
Evolution shrugged.
Basically. For things like lysine or tryptophan, our bodies effectively said, you'll probably eat enough of this. It's way too expensive to build a factory for it, so we'll just trust the grocery store. It's a calculated risk.
But for the ones we're discussing today.
No risk allowed. These are mission critical.
So the mission today is to tour that factory floor. We aren't looking at what we eat. We're looking at what we make.
We're analyzing the chemical assembly lines that are running inside your cells right now. We're going to look at how your body takes the scraps from burning sugar and turns them into the literal building blocks of life.
And we're talking about things like the glutamate family, the transamination station.
Yeah, and even some rule-breaking molecules that act like biological hackers.
And we should probably mention the stakes here. Because when I was reading the source material Rodwell's text, it became clear that when these homemade factories break down, it's not just a minor inconvenience.
No, absolutely not. When these enzymes malfunction, or when the raw materials run dry, we aren't talking about feeling a bit tired. We're talking about catastrophic system failures.
Like what?
We're going to discuss scurvy, which is actually a breakdown in this exact machinery. We'll look at kwashiorkor, the tragedy of weaning. We'll get into Menkes syndrome, where your hair literally turns to steel wool.
And there was something about a 21st amino acid, which I thought was a typo because I was taught my whole life there were only 20.
Not a typo. It's a biological special app. But we'll get there.
All right, let's start with the big picture then. Section 1 of our outline, the evolutionary economy of metabolism. You mentioned the body decided to make some and not others. What's the logic?
It comes down to cost, pure and simple. The body is a business. Every single time you build a molecule, you have to pay for it.
And the currency is ATP, right? Adenosine triphosphate.
That's the energy coin of the realm, yes. But there's another cost, which is arguably even higher, the cost of infrastructure. I want you to look at table 2722 in the source material. It breaks down the enzyme count.
Okay, I'm looking at it. It lists the enzymes required to synthesize the non-essential amino acids, the ones we make.
Just count them up. To maintain the ability to make these 12 amino acids, the human genome has to code for, maintain, and regulate 59 different enzymes.
59! That's a lot of overhead. You have to transcribe the DNA, fold the proteins, keep them working. It's a constant job.
It is a massive investment. But now, compare that to the essential amino acids, the ones we don't make. If we as humans wanted to make lysine or methionine or threonine and the rest, we would need hundreds of additional enzymes.
Hundreds?
Oh, yeah. The pathways in plants and bacteria that create those molecules are incredibly long and complex. They're multi-step, energy-hungry processes.
So, somewhere back in the swamp of evolutionary history, our ancestors did a cost-benefit analysis.
In a way, yes. It's a concept called survival value. Imagine a primitive, single-celled organism. It's spending, say, 30% of its total energy budget just building tryptophan factories.
Okay.
But it lives in a soup that is just full of tryptophan from other dead organisms.
So it's wasting money. It's building a factory to make water while living in a lake.
Exactly. Then a random mutation happens. One organism loses the genes to make tryptophan. It just can't do it anymore.
That sounds like a bad thing.
But think about it. Suddenly, it has 30% more energy available. It can swim faster, divide sooner, survive harsh conditions longer. That lazy mutant outcompetes the hard worker.
Wait, calling it lazy feels like we're judging. Isn't it just risky? What if the lake dries up? What if the food source runs out?
That is the trade-off. That is the fundamental bargain we made. We traded nutritional independence for metabolic efficiency.
So we became streamlined, high-performance machines.
Right. But the price we paid is that we are tethered to the food web. We are completely dependent on other organisms to do the heavy lifting for us.
We outsource the difficult chemistry to plants and bacteria.
We did. But for the 12 we're discussing today, the math went the other way. The pathways were short enough, and the molecules were so critical that we kept the machinery.
Okay, so we kept the factory for these 12. But a factory needs raw materials. You can't make protein out of thin air. Where does the carbon come from?
This is where the efficiency really shines.
Hmm.
We don't use exotic materials. We make these amino acids from amphibolic intermediates.
Amphibolic, good Scrabble word. What does it mean?
It comes from amphe, meaning both. These are molecules that are at a crossroads. They're involved in catabolism, breaking things down, like burning sugar for energy. But they can also be snatched up for anabolism, for building things.
So they can go either way.
Precisely. Specifically, we steal them from glycolysis and the citric acid cycle.
So we eat sugar. We start burning that sugar in the mitochondrial furnace, and then the amino acid factory just reaches into the furnace and pulls out half-burnt logs.
That is a surprisingly accurate analogy. We pulled out these carbon chains, alpha-keto acids, before they're fully burned into CO2, and we use them as the skeletons for our new amino acids.
But an amino acid isn't just a carbon skeleton. The name gives it away. It needs an amino group. It needs nitrogen.
And that brings us perfectly to section 2, the glutamate family. This is the nitrogen hub of the entire body.
Nitrogen is tricky, right? Because the nitrogen gas in the air is useless to us, and ammonia, that's toxic.
Toxic is an understatement. Ammonium ions, NH4 plus ReO, are profoundly cytotoxic. They interfere with the pH balance and the electrochemical gradients in your brain. If ammonia levels rise too high, you suffer tremors, slurred speech, coma, and eventually death.
So we have a paradox. We need nitrogen to build life, but the raw, usable form of nitrogen kills us.
Enter glutamate. This is the body's brilliant solution to the nitrogen paradox. Look at figure 27:1, the reaction for glutamate dehydrogenase.
Okay, I see the diagram. We have alpha-ketoglutarate on the left.
Alpha-ketoglutarate is one of those half-burnt logs from the citric acid cycle. It's just a 5-carbon chain floating around in the mitochondria. The enzyme grabs this chain.
And it grabs the dangerous ammonia.
It grabs a dangerous free-floating ammonium ion, yes, and it welds them together.
How?
It performs a reaction called reductive amidation. It uses electron power in the form of NADPH to force that nitrogen onto the carbon chain. And the result is L-glutamate.
So glutamate is basically a safe storage container for toxic ammonia.
It's a biological hazmat container, exactly. But it's not just for storage, it's for utility. Yeah. Once you have the nitrogen locked into glutamate, you can do things with it.
But the text mentions that sometimes you need to transport nitrogen around the body.
Right, from the liver to the kidneys, or from your muscles to your gut. And glutamate is negatively charged. It's bulky. It's not the best traveler.
So we need a better vehicle.
We upgraded to glutamine.
Glutamate versus glutamine. I feel like people mix these up constantly. What's the real difference?
The difference is just one nitrogen. Glutamine is glutamate plus another nitrogen group. It carries a double load.
It's efficient.
Very. But attaching that second nitrogen is chemically difficult. You can't just stick 2 repelling magnets together. You need to force them. This brings us to the enzyme glutamine synthetase and figure 27:2. This is a beautiful piece of mechanical engineering.
I see ATP in the mix here, so this costs energy.
It does.
Yeah.
And understanding how it uses ATP unlocks how the body drives so many chemical reactions. It doesn't just use energy like a battery. It's mechanical. It's physical.
Walk me through the mechanism, step by step.
Okay, step 1. The enzyme takes glutamate and it slams a phosphate group from ATP onto it.
So it literally rips a piece off the ATP and sticks it to the glutamate.
Yes. This creates a highly volatile intermediate called gamma-glutamyl phosphate. Think of this molecule like a mousetrap that you have just set. It is vibrating with tension. It is incredibly unstable. That phosphate group is desperately trying to leave because the bond is so strained.
It's a loaded spring.
It is a loaded spring. And just as it's about to snap, the enzyme brings in the ammonium ion. The ammonium sees that vulnerability and it attacks. The phosphate pops off snap and the ammonium takes its place.
Wow. So the energy from the ATP was used to set the trap?
Exactly. The release of that phosphate provides the thermodynamic push to make the bond happen. And because the phosphate just floats away, the reaction can't easily go backward. You have now created glutamine.
And now that we have glutamine, we have a safe, neutral molecule that can just float through the blood.
It's the Amazon Prime delivery truck of the nitrogen world. In fact, glutamine is the most abundant free amino acid in the human body.
Really?
By far. Because it is constantly shuttling nitrogen between organs. It's always on the move.
Okay, so we've secured the nitrogen, we have the hub. Now we need to make the other amino acids. This leads us to section 3, the transamination station.
Transamination is a fancy word for a really simple concept.
The Lego analogy.
It works perfectly. Imagine an amino acid is a Lego figure. You have the body, that's the carbon skeleton, the alpha-keto acid, and you have the head, that's the nitrogen group, the amino group.
And transamination is just swapping heads.
Precisely. You don't build a new head from scratch every single time. That would be inefficient.
Yeah.
You just take the head off glutamate, our hub, and you snap it onto a different body. This is exactly how we make alanine and aspartate.
Let's trace alanine first. Figure 27-4.
Okay. The body for alanine is pyruvate. Pyruvate is the end product of glycolysis. If you break a sugar molecule in half, you get pyruvate.
So you take pyruvate and you bring in a glutamate.
An enzyme called alanine aminotransferase, or ALT. You see this on blood tests. It grabs both. It pops the nitrogen head off glutamate, sticks it onto pyruvate, and boom, you have alanine.
And what's left of the glutamate?
It goes back to being alpha-ketoglutarate. Its head is gone. It can go back to the citric acid cycle to get recycled or burned for energy. It's a perfect circle.
And aspartate.
Same trick, different body. The body here is oxaloacetate.
Oxaloacetate, that's the molecule that restarts the Krebs cycle, right? It picks up the acetyl-CoA.
It is. It's the starter key for the whole cycle. You take oxaloacetate, you swap a nitrogen onto it, and it becomes aspartate. These reactions are totally reversible. They flow back and forth like tides, depending on what the cell needs at that moment.
But then we get to asparagine, figure 27-5. This looks like the glutamine situation all over again.
It is very analogous. Asparagine is the eminide of aspartate. Just like glutamine is the eminide of glutamate. We are adding a second nitrogen.
But the mechanism here has a twist. I'm looking at the diagram, and the ATP usage is different.
A very expensive twist. You have a good eye. In the glutamine reaction, we turned ATP into ADP. We used one high-energy phosphate bond. For asparagine synthesis, the enzyme asparagine synthetase breaks ATP all the way down to AMP, adenosine monophosphate, plus pyrophosphate, or PAI.
Breaking it down to AMP, that's like stripping off 2 phosphates. That's like paying with 2 coins instead of 1.
Effectively, yes. But here's the genius part. That pyrophosphate, the PPI that gets released, another enzyme comes along immediately and smashes it into 2 separate phosphates.
Why does that matter? Why the extra step?
It's a thermodynamic ratchet. Chemical reactions, especially in biology, can often go backward. But by immediately destroying one of the products, the pyrophosphate, the body effectively burns the bridge behind it. There is no PPI left to help reverse the reaction. It forces the production of asparagine to be a one-way street.
That seems aggressive. Why do we need to force it so hard?
Because asparagine is absolutely critical for cell signaling and for building glycoproteins. The body cannot afford to have this reaction fluctuate. It pays a premium in energy to absolutely guarantee supply.
It's amazing to think about the body managing this budget. Okay, for this one, we pay double, no questions asked. Let's move to section 4, the glycolysis connection. We talked about pyruvate, but there are other stops on the sugar-burning train.
Let's get off at the station called 3-phosphoglycerate.
Try saying that 5 times fast.
It's a mouthful. But this molecule is the grandfather of the serine family. This pathway, shown in figure 27-7, is like a masterclass in chemical sculpting. It happens in 3 distinct, elegant steps.
Okay, paint the picture for me. Step 1.
Step 1 is oxidation. We have this 3-phosphoglycerate. An enzyme called a dehydrogenase comes in, and it steals 2 hydrogen atoms, really, the electrons, from the molecule.
So it changes the shape?
It fundamentally changes the chemistry. It turns an alcohol group, an OH, into a ketone, which is a double-bonded oxygen. Now, the molecule is called 3-phosphohydroxypyruvate. It has been prepped. It's ready to receive a nitrogen.
Step 2.
Transamination. We bring in our friend glutamate again. We do the head swap. We pop the nitrogen head onto this prepped molecule. Now it is called phosphoL-serine.
So it's almost serine, but it still has that phospho part attached, that phosphate group.
Correct. It's still wearing its seatbelt. So step 3 is hydrolysis, or more simply, dephosphorylation. A phosphatase enzyme comes in with a pair of scissors and just cuts the phosphate bond. The seatbelt falls off, and you are left with free serine.
Serine is huge in skincare commercials, but inside the body, it's a precursor, right? It's used to make other things.
It is a major hub. From serine, we make glycine and cysteine. Let's talk about glycine for a moment.
The simplest amino acid.
Structurally, yes, it's tiny. It has no fancy side chain, just 2 hydrogen atoms. But its importance is inverse to its size. It's used to make heme for your blood, purines for your DNA, and of course, collagen.
The outline mentions 3 different routes to make it. Why the redundancy?
Because it's so critical. You need backup plans. Route A is the main one. It comes directly from serine. The enzyme serine hydroxyamyltransferase does something remarkable. It cuts the side chain off serine.
It just chops a piece of the molecule off?
It transfers a single carbon unit from serine to a helper molecule called folate, specifically tetrahydrofolate.
Wait, folate, as in folic acid, the stuff they tell pregnant women to take.
Exactly. This is precisely why folate is essential in our diet. Folate is the body's carrier of 1 carbon units. When we make glycine from serine, we are simultaneously charging up our folate with a carbon unit. That charged folate then goes off to help build DNA.
This connects so many dots. So if I'm deficient in folate,
You jam up this whole pathway. You can't make glycine efficiently, and more importantly, you can't charge up the folate you need for DNA synthesis.
Right.
This is why folate deficiency causes neural tube defects in developing embryos. They are trying to build DNA at an explosive rate, and the machinery stalls.
it's not just about the vitamin itself, it's about the machinery the vitamin serves.
It's always about the machinery. And just to mention, route B involves breaking down choline-like from eggs or liver in a salvage pathway. The body hates waste. If you have old choline, it will strip it down to parts and make glycine.
Okay, this brings us to section 5, which I think is the most intellectually satisfying part of this whole chapter. The cheat amino acids,
Cysteine and tyrosine. I like that name for them.
we call them non-essential, which, as we've learned, means we make them. But the fine print is we can only make them if we eat other specific essential amino acids first.
It's a paradox, isn't it?
Yeah.
It's like saying, oh yeah, I can make a grilled cheese sandwich from scratch as long as you give me the bread and the cheese.
You're just assembling it. You're not
I'm like,
making
well,
it.
That's exactly what the body is doing. Let's look at cysteine. Cysteine contains sulfur. It's a sulfur-based amino acid. But humans,
Yeah.
we are not nuclear reactors. We can't perform fusion. We cannot turn carbon into sulfur.
So where do we get the sulfur?
We have to eat it. And the primary source of dietary organic sulfur is an amino acid called methionine. Methionine is an essential amino acid.
So to make cysteine the non-essential one, we need methionine, the essential one. How does that work?
The pathway is called the methionine cycle, and you can see it in figure 2711. It's a bit of a heist movie. First, methionine gets processed into a molecule called homocysteine. Then this homocysteine meets up with serine, which we just learned how to make from sugar. They merge. An enzyme joins them together to form a massive Frankenstein molecule called cystathionine.
A merger.
Yes. But then, an enzyme called cystathionine lyase steps in to perform the heist. It chops the molecule in half, but here's the trick. It doesn't chop it where they joined. It chops it in such a way that the sulfur atom, which originally belonged to methionine, stays with the serine skeleton.
The serine stole the sulfur.
They walked away with the goods. The original methionine skeleton, now called alpha-ketobutyrate, is discarded or recycled. And the serine with sulfur is now called cysteine.
That is brilliant, but it means that if I don't eat enough methionine,
You cannot make cysteine, period. This is why in nutrition we say cysteine has a sparing effect on methionine. If you eat cysteine directly, like from whey protein, you don't need to burn as much of your precious methionine to make it.
and the same logic applies to tyrosine.
Tyrosine is even more direct.
Yeah.
Tyrosine is synthesized from one molecule, phenylalanine, and phenylalanine is essential.
Phenylalanine. I see that warning on cans of diet soda. Phenylketonuria contains phenylalanine.
We're going to explain exactly why that warning exists. But first, let's look at the healthy mechanism. The enzyme is phenylalanine hydroxylase. It takes phenylalanine, which has a simple ring structure, and it uses molecular oxygen to staple an OH group, a hydroxyl group, onto that ring.
Just one oxygen atom makes the difference.
One oxygen changes everything. Phenylalanine is hydrophobic. It hates water. Tyrosine, with that OH group, is hydrophilic and much more reactive. It becomes the precursor for dopamine, epinephrine, norepinephrine, all your major stress and reward neurotransmitters.
So one single enzyme stands between the raw material from my diet and my brain chemicals.
Correct. Now let's ask the critical question. What happens if that enzyme is broken? What if you were born with a genetic mutation in phenylalanine hydroxylase?
We can't make tyrosine.
That's problem number 1. But problem number 2 is much worse. The phenylalanine starts to pile up. It has nowhere to go. The concentration builds in the blood and it crosses the blood-brain barrier. At high levels, phenylalanine is a potent neurotoxin to developing neurons. It causes severe, irreversible intellectual disability. This is phenylketonuria, or PKU.
And that's why they prick the heel of every newborn baby in the hospital.
Exactly. The Guthrie test. They are testing for high phenylalanine levels. If they catch it on day 1, they can put the baby on a special synthetic diet, a formula that's extremely low in phenylalanine, but high in tyrosine.
And the kid grows up normal.
Completely normal. It is one of the greatest miracles of modern biochemistry. We turned a devastating genetic curse into a manageable dietary issue. But notice something important. For a person with PKU, tyrosine becomes an essential amino acid. They cannot make it. They must eat it.
It really blurs the lines of these definitions. Essential isn't a fixed property. It depends on your own personal genes.
It depends on your machinery.
Speaking of machinery and structure, let's move to Section 6, the structural specialists. Proline and the whole collagen story.
Proline is the oddball of the group. Look at figure 2710. Most amino acids look like a key, a central carbon with a side chain sticking out. Proline curls around and bites its own tail.
It forms a ring.
It forms a rigid, five-membered ring that actually includes its own nitrogen. This makes proline extremely stiff. In a protein chain, proline acts like a kink or a hard turn. It's absolutely crucial for creating specific three-dimensional structures.
And the biggest structure in the human body is collagen.
Skin, bones, tendons, ligaments. It's the rebar, the glue that holds us together.
Yep.
But here is the fascinating twist. Collagen is full of two amino acids that are not in the genetic code: hydroxyproline and hydroxylysine.
Wait, if they aren't in the DNA code, how do they get into the protein?
Post-translational modification, we have to retrofit the protein after it's been built.
Retrofit, like adding a spoiler to a car after it leaves the factory.
That is a perfect analogy. The ribosome builds a procollagen chain using normal proline and normal lysine. It prints the standard model. Then, once that chain is built, a crew of enzymes, prolyl hydroxylase and lysyl hydroxylase, crawl along the chain. They find specific prolines and lysines and attach an oxygen group to them.
Why bother? What does that extra oxygen do?
It acts like chemical Velcro. The hydroxyproline allows the three separate strands of collagen to bond tightly together into a stable triple helix. Without it, the helix is unstable. At body temperature, normal collagen would just unravel.
It would melt.
Effectively, yes.
Yeah.
It would turn into gelatin. And this brings us to the history lesson. These retrofitting enzymes, the hydroxylases, are very needy. To work, they require iron. But not just any iron. It has to be in the reduced state, F2 plus Leyros.
Okay, chemistry flashback. Iron oxidizes easily. It rusts. It turns into F3 plus layer.
Right. And every time the enzyme works, there's a risk. The iron at its core oxidizes and the enzyme jams. It breaks. To reset the enzyme, to essentially clean off the rust and get it working again, you need a reducing agent. You need ascorbate.
Vitamin C.
Vitamin C. Its primary job in this context is to keep those hydroxylase enzymes running so you can manufacture a hydroxyproline and make stable collagen.
And this is why scurvy happens.
Yes, and here is the evolutionary kicker. Most animals, dogs, cats, cows, mice, they make their own vitamin C. They have a gene called GLOO that synthesizes it from glucose. They can never get scurvy.
But we don't. Humans, primates.
We have the GLOO gene. It is sitting right there on chromosome 8. But it's broken. It's a pseudogene.
Yeah.
It has a massive mutation that disabled it about 60 million years ago in our primate ancestors.
Why? Why on earth would we lose such a useful gene?
The leading theory is that our ancestors were fruit eaters living in the tropics. They were getting so much vitamin C from their diet that the gene became redundant. It was a classic case of use it or lose it. The mutation happened and it didn't kill them, so it stuck.
It didn't kill them then. But fast forward to a British sailor in 1750, stuck on a ship with only dried meat and hardtack.
And suddenly, that neutral mutation becomes a death sentence. Without vitamin C, the prolyl hydroxylase stops. You stop making hydroxyproline. Your new collagen synthesis grinds to a halt. Your existing collagen starts to unravel.
And the symptoms
are literally your body falling apart at the seams, bleeding gums because the capillaries explode, teeth falling out because the ligaments holding them in dissolve, and the most horrifying one, old wound scars you've had for 20 years suddenly break open and start bleeding again because the repair tissue disintegrates.
That is absolute nightmare fuel, all because of a single missing hydroxyl group on a single amino acid.
It shows how fragile our supposed independence really is.
Okay, from the fragility of the past to the weirdness of the present. Section 7, the 21st amino acid.
Selenocysteine.
I really thought the genetic code was a closed system. 20 amino acids. That's it. Done.
Biology just loves to break its own rules. Selenocysteine is a rare special forces amino acid. It's found in the active sites of just a few dozen enzymes in humans, but they are incredibly critical ones.
What makes it so special?
The name gives it away. Instead of sulfur, like in cysteine, it uses selenium. Selenium is a semi-metal. It sits just below sulfur on the periodic table. It's much more reactive. It allows enzymes to perform antioxidant reactions, fighting free radicals, at speeds that sulfur just can't match.
But how do we make it? The outline suggests it breaks all the normal rules of synthesis.
It does. It's completely unique. Normally, a tRNA, the delivery truck, picks up a finished amino acid and drives it to the ribosome. For selenocysteine, the synthesis happens on the truck.
What? You're kidding.
I am not. Look at figure 2714 first. A specific special tRNA is charged with serine, a normal serine.
Okay.
Then, an enzyme called selenophosphate synthetase brings in a selenium atom, which we get from our diet. Another enzyme then converts that serine into selenocysteine while it is still attached to the tRNA.
It modifies the package while it's in the FedEx truck.
Exactly. But the delivery address is even crazier. The genetic code, the codon on the mRNA for selenocysteine is UGA.
Wait, no. I remember my biology 101. UAA, UAG, and UGA are the stop codons. They tell the ribosome to quit, terminate the protein.
In 99.9% of cases, yes. But for these specific selenoproteins, there is a hack. The mRNA contains a special structure further down the chain called a SESIS element, a selenocysteine insertion sequence. It's a little stem loop structure.
Stem loop.
Imagine a specific complex knot in the ticker tape. When the ribosome hits the UGA codon, it pauses. It looks ahead and it sees this knot. The knot interacts with a special elongation factor that effectively says, hey, I know this looks like a stop sign, but ignore it. It's actually a code for selenocysteine. Insert the selenium and keep building.
It's a contextual code override.
It is biological hacking at its finest, and we desperately need it. Without selenocysteine, enzymes like glutathione peroxidase don't work. We lose one of our primary defenses against oxidative stress.
And that leads to disease, like Keyshawn disease.
Yes, a fatal cardiomyopathy, a heart condition that was found in parts of China where the soil is completely devoid of selenium. The heart muscle literally dies because it can't protect itself from oxidation without this 21st amino acid.
This brings us to the final section, nutritional disorders and clinical implications. We've touched on scurvy, PKU, Keyshawn, but let's talk about the big ones you see in global health, kwashiorkor and marasmus.
These are the diseases of protein-calorie malnutrition. They're heartbreaking, but from a biochemical standpoint, they teach us a lot about the metabolic priorities of the body.
Kwashiorkor is the one with the distended belly, right? I've seen the picture.
Yes. The name comes from the Ga language of coastal Ghana. It translates roughly to the thickness the baby gets when the new baby comes.
That is profound. It's about displacement.
It's about weaning. When a new baby arrives, the older child, maybe 1 or 2 years old, is taken off of breast milk. Breast milk is a perfect protein source. They're switched to the family diet, which in many parts of the world is starchy gruels made from cassava, yam, or rice.
So they are getting calories. They have plenty of energy from the start.
You have plenty of glucose, but they have almost no amino acids. Their factory has fuel, but it has no building materials, no nitrogen.
What happens physiologically?
The liver tries to keep working, but it can't synthesize the proteins it needs to export. Specifically, it can't make albumin. Albumin is the most abundant protein in your blood. Its main job is to maintain osmotic pressure. It acts like a sponge to keep the water inside the blood vessels.
So if your albumin level drops,
the water leaks out. It leaks into the abdominal cavity. That causes the classic distended belly, which is called ascites. It also causes swollen feet, which is edema. At the same time, the liver becomes fatty because it can't build the lipoproteins needed to ship fat out of the liver.
So the child looks plump in the belly, but they are literally starving for nitrogen.
Exactly. Now contrast that with marasmus. Marasmus is total starvation. It's a deficiency in calories and protein.
That's the skin and bones look.
Yes. In marasmus, the body switches into a desperate survival mode. It catabolizes, it eats its own muscle tissue and fat to get the amino acids and energy it needs to keep the brain and heart alive. It's tragic, but in a strange way, it's a more balanced form of starvation than kwashiorkor.
Lastly, Menkes syndrome. You mentioned kinky hair.
This circles back perfectly to our factory inputs. We talked about vitamin C being a cofactor. Menkes is about copper.
Why do we need copper? I thought that was for wires.
We need it for enzymes. Remember lysyl hydroxylase for making collagen? Well, there's another enzyme called lysyl oxidase. It works outside the cell to cross-link the collagen and elastin fibers. It's the enzyme that welds the steel beams of your tissues together to give them strength.
And it runs on copper.
Copper is its essential cofactor. In Menkes syndrome, there's a genetic defect in a copper transporter protein, so the gut can't absorb copper from food. No copper means no lysyl oxidase, which means no cross-linking.
And the hair, what does that have to do with it?
Hair is made of keratin, which is heavily cross-linked with disulfide bonds. But other copper-dependent enzymes are involved in its formation and structure. Without them, the keratin structure fails. The hair looks twisted, brittle, and silvery under a microscope. The classic sign is called pili torti. But the real damage, The life-threatening damage is in the arteries and the brain, which are losing their structural integrity.
It's just incredible. We think of ourselves as these autonomous, robust beings, but we're really these delicate chemical factories, completely dependent on trace amounts of copper, selenium, vitamin C, and specific amino acids from our diet.
That is the ultimate takeaway of this deep dive. We draw this sharp line between nutritionally essential and metabolically essential. But biologically, all 20 or 21 amino acids are absolutely essential. You cannot live without any of them. The only difference is the paperwork.
The paperwork.
Who does the paperwork of making them? For 12 of them, our bodies do the paperwork. We run the factory. For the other 8, we let the plants and bacteria do the paperwork, and we just collect the finished product at the dinner table.
Outsourcing the paperwork. I like that. But it really does highlight our vulnerability.
It does. A single missing input like vitamin C halts a critical assembly line. A single genetic typo like in PKU or Menkes causes either toxic waste to pile up or leads to total structural collapse.
It makes you look at a multivitamin or a piece of steak or a handful of nuts completely differently. You aren't just eating food, you are restocking the raw materials for a factory that never ever sleeps.
Precisely. And in a modern world where we are often overfed but undernourished, understanding these specific micronutrient requirements, folate, selenium, copper, is more important than ever.
So here's a thought to leave you with. Evolution decided it was smarter to lose the ability to make those 8 amino acids just to save a little energy. It was a bet. It was a bet made millions of years ago in a world of scarcity, but in our modern world of abundance.
We're starting to see the cracks in that strategy.
Exactly. Are we now vulnerable because of an evolutionary bet that paid off for a primate in a jungle, but might not pay off for a modern human eating processed food? Something to chew on. Thanks for joining us on this deep dive into the chemical factory of you. Go eat some protein. We will catch you on the next one.
Stay curious.